We'll start with just a quick review of membrane transport biochemistry, and then we'll go through some practice problems. The key thing I want you to be able to do is to be able to recognize and classify different types of membrane transport, classify them as passive or active, further subclassify them as we'll talk about, and see if they are co-transport, unitransport, and if co-transport, symport, or antiport. So let's go through these terminology. Starting with how we can differentiate between active and passive transport, the key really is thinking about whether something is going along or against its electrochemical gradient. So the electrochemical gradient is the combination of a chemical gradient where you're thinking about molecules going from where they have a high concentration to where they have a low concentration, and an electrical gradient where if charged molecules are going to move towards the opposite charge. Moving along your gradient, so going from positive to negative or negative to positive, or going from high concentration to low concentration, that's going to be energetically favorable. The larger the gradient that you're moving along, the more energy you're going to release. However, there's going to be an equal and opposite energy in the other direction. So the further you are uphill, the more energy you're going to get sliding downhill, but the more energy it'll take to walk back uphill. If a molecule is going down its electrochemical gradient, remember that's going to be energetically favorable. It's going to have a negative delta G. It's going to give off energy. It's not going to need energy in order to do so, but it might need a little help. And so passive transport is when we're going along a gradient. So we don't need energy, but we might need a door, kind of. So if we think about membranes, we have this hydrophobic interior, these phospholipids with these hydrophilic heads, so they can stick out towards the aqueous solutions. And then they have these hydrophobic in middles. It's like your sandwich. Molecules need to get through that sandwich in order to get through the membrane. And if a molecule is hydrophilic, so it'll be good to hang out with these aqueous solutions, it's going to have a hard time getting through the membrane. And so some molecules are small enough, they're nonpolar enough that they can kind of just sneak through. An example is like our steroid hormones, as well as even water because it's small enough. And but not something small things like ions, those aren't going to go through because they're too charged. But those small molecules that can just sneak in through, we call that simple diffusion. However, a lot of molecules are going to be wanting to go along their gradient, but they just can't get through the membrane because it's too nonpolar. And so then we have facilitated diffusion where we have proteins that act as channels, selective channels typically, that are going to let selective molecules through. But the molecules are traveling along their gradient. They're going from high to low concentration, and sometimes they're going from their charge to the opposite charge. And remember, it's the combination of the two, the electrochemical gradient, that's going to dictate which way is favorable. But then whether you can actually go through is going to dictate whether there's a passage for you. These passages are often going to be gated, and so they're only going to open up in response to a stimulus. This is going to allow you to build up a gradient that then you can use, you can let through when you're ready, rather than just having the gradient dissipate itself out. Because remember, the gradient is what provides the drive to move. If you let things flow freely, then you're going to dissipate your gradient. So passive transport is when molecules are moving along their electrochemical gradient. So they're moving in a direction that is thermodynamically favorable. They can do it either with help in the case of facilitated diffusion, when we're talking about channels, often selective channels, or simple diffusion, where it's molecules that are small and nonpolar enough that they can just sneak through the membrane without help. Contrast that with active transport. With active transport, you're moving against your electrochemical gradient. So if you're moving against your electrochemical gradient, somehow you're going to have to be spending energy. Sometimes you spend this energy directly at the site of the transport protein that's going to let you through. This is what we call primary active transport. Often you're going to be doing something like hydrolyzing ATP, sometimes directly or sometimes by phosphorylating the pump. So we often call it a pump when we have a primary active transporter. It's going to be hydrolyzing ATP, or it's going to be phosphorylating itself, and that'll get it to change shape in such a way that it's going to open up the channel and let the molecules through. The molecules are going to be traveling against their gradient, so you're using the energy from that ATP or from that other high, quote unquote, high energy compound. They're using that energy in order to transport the molecule. So we often call these pumps, and you're spending the energy directly at the site where you're going through, where that molecule is going through. Don't confuse that with secondary active transport. So in secondary active transport, you're still going to require energy. You're still actually going to be using primary active transport, but you're going to be using that to create a gradient that's going to pay ahead for the cost of bringing another molecule in. So you use primary active transport to create a gradient of one molecule. You then let that molecule flow back along its gradient, which now is going to release energy, and the energy that's released is going to be used to transport another molecule along with it. This is going to involve a form of co-transport because you have two molecules traveling together, one along its gradient that's releasing energy, and one against its gradient that's requiring energy. Let's look at an example. In your kidneys, you have these tubules, and they're going to be responsible for helping you control your blood glucose and say what goes into your blood versus what goes in your urine. And there's this whole reabsorption thing, and I'm not a physiologist, so I'm not going to get too much into try to get into that, but we will look at some of the membrane transport that is involved. So we have these renal proximal tubules, and then on one side you have the urine, and then on the other side you have the blood. We want to take glucose from the urine and reabsorb it back into the bloodstream. Inside of our tubules, so inside of these tubules, we have a high concentration of potassium and a low concentration of sodium. And then in our urine, we have a high concentration of sodium. And then in our blood, we have a high concentration of sodium and a low concentration of potassium. We also, in our tubules, have a high concentration of glucose. And then on either side, in our urine and in our blood, we have a lower concentration of glucose. So we want to think about how we can get glucose from the urine, where it has a low concentration, into the tubules, where it had a high concentration. So here we're going to have to go against the gradient and then go along the gradient in order to get it back into the blood. And so how are we going to do this? Let's think about these different transporters that are involved. So let's look at this GLUT2. This question is asking us what two types of transport is GLUT2 mediated glucose transport? And so we're going to have to identify it as either like co-transport, uniport, those sorts of terms, as well as active and passive, and then the subclassification. So whether it's facilitated diffusion, simple diffusion, primary active, or secondary active. So let's think about this GLUT2, and we are transporting glucose. So we're transporting a single thing, so that's going to be uniport. So there's one classification down. Now let's think about, are we going along its gradient, in which case it would be passive transport, or against its gradient, in which case it would be active transport. In this case, what we have is that we have the glucose is going from high glucose to low glucose, so that's going to be passive transport. We can see, though, that it's going through a channel, so that's going to be facilitated diffusion. Okay, now let's take another one. SGL2. SGL2 is right here, and let's look at what we can tell from this diagram. We see that sodium is going from the urine into the renal tubules, and glucose is going from the urine into the renal tubules. In this case, glucose is going from a low concentration to a high concentration, and sodium is going from a high concentration to a low concentration. So sodium is going along its gradient, whereas glucose is going against its gradient. So this is going to be releasing energy that's used to power, to give energy to this energy requiring transport of the glucose. This is an example of secondary active transport. So we had to somehow have primary active transport that was keeping the sodium gradient that we can then use to bring in the glucose. And so we'll look at that in a minute. But first, let's also classify it as it's based on how many molecules it's transporting and in what directions. So we're transporting two molecules. So this is co-transport, and they're going in the same direction. So this is going to be symport. So let's go back and think about secondary active transport. So how are we keeping this gradient of the sodium? Well, here we need to look at what's going on at this side. We have this ATPase. Now this ATPase, what it's doing is it's going to be taking sodium from the tubules and into the blood. So it's going to be going from low sodium to high sodium. And so in this case, we're going against the gradient. We're pumping the sodium against its gradient. And let's look at the potassium. We're going to go from low potassium to high potassium. So we're pumping that against its gradient. And so in this case, what we do is we have two molecules that are moving against their gradients. So we're going to have to be spending energy directly. This is going to be a pump. This is going to be primary active transport. And the significance of doing this is that we're then keeping the sodium concentrations in these tubules low so that we maintain this gradient of the sodium, this favorable gradient that we can use then to power the symport through secondary active transport of the glucose. So when we think about secondary active transport, you basically have one molecule that's doing facilitated diffusion to power the active diffusion of a second molecule. And so it would be active in the sense of thinking about the glucose because the glucose is moving from low concentration to high concentration. Okay, now let's think about what would happen if we perturb the system. And so I just took this figure from like online, which is why it's kind of weird, and I have to like annotate it to make it a little clearer. But they were thinking about what would happen if you added an SGL2 inhibitor. And so if you added this SGL2 inhibitor, and remember that the sign is kind of what we often see. If we see an inhibitor, we're going to see something like red. We're going to see one of these T signs, maybe a minus sign. In this case, this SGL2 inhibitor is going to be inhibiting this SGL2 channel. And remember that was going to be powering the secondary active transport of the glucose using the gradient of the sodium. And the gradient of the sodium was kept by the pumping of the sodium out of the cells through this ATPase. And if we were to inhibit this, then the glucose can't get through. And so then the glucose in the urine is going to build up. What about our blood glucose? Well, if we can't get glucose into the tubules, we can't get it into the blood. So our blood glucose is going to go down. Now let's think about our urine sodium. Well, our urine sodium, it was coming through with the glucose. If you had an SGL2 inhibitor, well, now the sodium can't go back through. If the sodium can't go back through, then it's going to stay in the urine, and that increases the levels in the urine. Note too that you would have similar results if you had low glucose in your urine. If you have low glucose in your urine, well, now there's nothing to let the sodium in. The sodium, this channel is only going to open when they're both present to go through, so that you're not basically wasting a ride. So it's like you don't want to have your empty bus running if all you have is the driver. So the sodium is kind of like the driver. You don't want the driver to be going if there's no passengers around. And so these channels are going to open up and when both things are present. But if you don't have one of them, then the other's not going to go. And so you need both the sodium and the glucose in order to get this to go.